Okay, in this video I want to work another problem involving protein structure, and this was something I actually already recorded in a previous video, but it ran a little long, so I'm going to redo it here. And the question says, most globular proteins, when briefly heated to 65 degrees Celsius, denature. Explain why. So kind of a simple question, just asking us to explain why these globular proteins denature when heated to 65 degrees C. Well, I mean, if we think about it for just a quick second, we'll remember that proteins are largely held together by weak attractive forces. So weak intermolecular forces is basically what I'm trying to say here. So that's exactly what I will say. So I'll say proteins are largely held together by weakly attractive forces. And in this case, I'll say such as hydrogen bonds and hydrophobic interactions. So, proteins are largely held together by weakly attractive forces such as hydrogen bonds and hydrophobic interactions. And they said we're heating this to 65 degrees Celsius. So what I'll say next is essentially heat is energy. And we know this. Heat is energy. So there's enough energy in heating to 65 degrees C to break hydrogen bonds, or to disrupt hydrogen bonds is what I should say. I mean, break is not really necessarily the right word because they're not covalent bonds, so we're not actually breaking them. So I'll say heating to 65 degrees C provides enough energy to disrupt hydrogen bonds. So heating to 65 degrees provides enough energy to disrupt hydrogen bonds. And what I should say is, since the molecules are moving faster, it will be more difficult for the atoms to orient themselves properly to form hydrogen bonds. So let's take a look at what we have here so far, and let's read it back and see how it sounds. So proteins are largely held together by weakly attractive forces such as hydrogen bonds and hydrophobic interactions. Heat is energy. Heating to 65 degrees C provides enough energy to disrupt hydrogen bonds. Since the molecules are moving faster, it will be more difficult for the atoms to orient themselves properly to form hydrogen bonds. So the faster moving molecules will have a difficult time orienting themselves well enough to form the hydrogen bonds. That's exactly what I'm trying to say here, is that because there's more motion, it's going to be more difficult to form a hydrogen bond. And hydrophobic interactions, so hydrophobic interactions are at risk for the same reasons. So they're at risk for the exact same reasons. Hydrophobic interactions are at risk for the exact same reasons. So here's the second question here. now that we've answered the first one. It says globular proteins that contain multiple disulfide bonds often must be heated to higher temperatures before they denature. Explain. So globular proteins that contain multiple disulfide bonds often must be heated to higher temperatures before they denature. Explain why. So, if you remember from one of my previous videos, I actually talked about this, and one of the things I said is that disulfide bonds are covalent bonds. So what does this mean to us? Well, these are actually real chemical bonds. They are covalent bonds. And how does that affect what we're saying here? Well, covalent bonds are orders of magnitude stronger than the attractive forces holding proteins shape. So disulfide bonds are covalent bonds. Covalent bonds are orders of magnitude stronger than the attractive forces holding protein shape. And therefore, they just require significantly more energy to disrupt. So they require significantly more energy to disrupt. That's about all you'd have to say. So disulfide bonds are covalent bonds, and covalent bonds are orders of magnitude stronger than the attractive forces holding proteins together. When I talk about the attractive forces, what I'm talking about is hydrogen bonds, hydrophobic effect, etc. And they require significantly more energy to disrupt. So it's kind of a really simple and straightforward concept. Thanks.